Module 5, Objective 4, explain the transmembrane potential in terms of electrical and chemical gradients. All cell membranes produce electrical signals by the movement of ions. Membrane potential is particularly important for neurons since they're charged with transmitting electrical impulses. If you were to consider an average neuron, you have a cell body, you have dendrites, which are the short extensions off of the cell body, and then we have an axon, which is a longer extension off the cell body, and it tends to end in axon terminals. The resting membrane potential of a neuron is the membrane potential of a resting cell. From the resting membrane potential, we experience graded potentials. These are temporary, localized changes in the transmembrane potential that are caused by some type of stimulus, and we'll see examples of stimuli shortly. If graded potentials are strong enough, they will convert into an action potential, and it's the action potential which is the electrical impulse, the electrical signal that travels down the length of the axon. So they're produced by graded potential if the graded potential is strong enough, and they're propagated down the length of the axon to the synapse. Here we see just a figure of a generalized neuron. We can see the resting potential. It's sort of localized within the cell. The graded potential, which is the temporary localized change in the transmembrane potential, occurs here at the axon hillock. And the action potential is propagated down the length of the axon to the synapse. In addition to resting, graded, and action potentials, we have two other activities that are common. The synaptic activity dictates what is happening at the synapse, and information processing is how that postsynaptic cell responds. So we have two cells here. We have a postsynaptic cell, post meaning behind or after the synapse, and we have the presynaptic cell, in this case a neuron. So if we're focused on this particular synapse here, this cell is the presynaptic cell, and this cell is the postsynaptic cell. And that can easily change downstream, and we'll see examples of that later. In fact, let's just sort of represent it now. If I were to draw a series of neurons, and I'll just briefly create 3 neurons, if we are referring to this synapse, this becomes the pre, and this becomes the postsynaptic cell. But if we instead are referring to this synapse, this is the pre, and this becomes the post. So it's all relevant to which synapse we're discussing. The resting membrane potential, there's 3 concepts to consider. The first one is deals with the fluids. So the extracellular fluid, which is the fluid outside the cell, and the intracellular fluid, the fluid within the cell, they differ in terms of their ionic composition. Extracellular fluids contain high concentrations of sodium and chloride ions, while the cytosol contains high concentrations of potassium ions, as well as large negatively charged proteins. So we have a difference in the ionic concentration inside compared to outside of the cell, where sodium and chloride are all localized outside, at least predominantly, and potassium and negatively charged proteins are localized inside of the cell. The second factor to consider is the fact that cells have selectively permeable membranes. So remember, selectively permeable refers to the fact that these cells select or allow certain substance in or out of the cell while restricting the movement of others. And the third point or concept is that membrane permeability varies by ion. So some membranes may allow sodium more access than potassium, for example. So there is a variability within the membrane permeability based on the ion. So let's take a look at what we have here, and we'll note the membrane here. So here we have the plasma membrane of the cell. So this out here is the extracellular, while this is the intracellular fluids. And as we noted previously, there is a high concentration of sodium ions outside of the cell and a high concentration of potassium ions inside the cell. The concentration of intracellular potassium is high, and these ions tend to move out of the cell through leak channels. Potassium leak channels, as we see right here, are channels that are just constantly leaking potassium. So potassium is going to move out periodically. In addition, sodium ion concentration is relatively high outside in the extracellular fluid, and they're going to move into the cell through sodium leak channels. And the movement of these ions, whether it's potassium or sodium, is really dependent on the concentration gradient or the chemical gradient. Remember that everything desires a state of equilibrium. So if you have a lot of potassium outside of the cell, it wants to equilibrate. It wants to move inside of the cell. And these sodium ions are going to be moving in through these sodium leak channels. In the same fashion, potassium ions are highly concentrated inside of the cell, and they too want to reach a state of equilibrium. So they're going to move through potassium leak channels outside of the cell in order to reach a state of equilibrium. If this system were allowed to reach equilibrium, we would have equal numbers of sodium outside of the cell compared to in, and equal numbers of potassium inside of the cell compared to out. The mechanism that maintains this distribution, where most sodium are outside of the cell and most of your potassium ions are inside of the cell, is due to the sodium-potassium exchange pump. And we see that right here in the middle, sodium-potassium exchange pump. And as the name implies, it's going to exchange sodium for potassium. It's going to physically pump sodium out while bringing potassium into the cell. And this is an active process. Notice the utilization of ATP. So this requires activity. So this pump is constantly working in order to maintain a disequilibrium. And it's because of the constant use of this ATP that we establish warm-bloodedness. This is outside of the scope of this course, but we are warm-blooded animals. Well, how do we maintain temperatures above the environment? Well, the answer is because of all these leak channels. This sodium-potassium exchange pumps are constantly having to work in order to create this disequilibrium, and in doing so, they utilize a tremendous amount of ATP. And we know that ATP is made by largely by the mitochondrion, and that a lot of that energy is lost as heat. So we generate a lot of heat 24/7 as our sodium-potassium exchange pumps are constantly working in order to create this disequilibrium. There are a couple other factors to consider if we look at the right side of this image. The first one is passive electrical gradients. Potassium ions are going to leave the cytosol more rapidly than sodium ions enter. And this is because the plasma membrane is much more permeable to potassium than it is to sodium. As a result, there are more positive charges outside the plasma membrane uh, and negatively charged protein molecules within the cytosol. So there's more negative charges inside the cytosol. And because of this, you get an electrical gradient across the plasma membrane. If you were to take a probe, as we did right here, and place it within the cell membrane, we can actually measure the electrical charge of the inside of the membrane relative to out, and we can see that this gauge gives us a reading of negative 70 millivolts. And that's going to become important in just a minute. In fact, negative 70 millivolts is the average resting membrane potential of a cell. So that number is something to commit to memory. It's going to come up again, but all that's saying is that the inside of the cell is negative relative to the out, and we can actually quantify that, and if we do, we get a negative 70 millivolt difference. So the passive processes that act across the cell membrane create two gradients. The first gradient is the chemical gradient, and this is mainly due to the concentration of ions. If you were to consider an axon, there's an axon, we know that sodium is localized outside of the cell and potassium is localized inside of the cell. And remember, what keeps sodium out and potassium in is that sodium-potassium exchange pump that is constantly working. What would happen if that sodium-potassium exchange pump stopped working? Well, these two ions would reach a state of equilibrium. They would move through their leak channels, their respective leak channels, until sodium concentration outside of the cell is equal to that of sodium concentration inside of the cell. And likewise, potassium would also leak out of the cell until the two sides are equal. So both of these ions desire equilibrium, and that desire is going to drive sodium into the cell and potassium out, and that is the chemical gradient. In addition, we have an electrical gradient. Remember that the inside of the cell is negative 70 millivolts compared to out. So there is overall a negative charge inside compared to out. Sodium is a positive ion. What drives sodium in partially is that electrical gradient. So sodium, which is positive, is highly attracted to the negative 70 millivolt inside of the cell, and that's going to help thrust or force, push, pull, whatever you may have. It's going to cause sodium ions to flood into the cell. Likewise, potassium is positive, and we're going to see later how changes in this transmembrane potential helps to drive potassium out. So there's two gradients that exist within a resting cell, both chemical and electrical gradients, and we'll see how these operate as we move forward. So the equilibrium potential. Membrane potential at which there is no net movement of a particular ion across the cell is known as the equilibrium potential. And for that to occur for potassium, that equals negative 90 millivolts, and for sodium, it's positive 66 millivolts. So the plasma membrane is highly permeable to potassium, and that's going to explain the similarities of equilibrium potentials for potassium and the resting membrane potential of about negative 70. The resting membrane's permeability to sodium is not high. In fact, it's very low. So sodium has a small effect on the resting membrane potential. So we've already noted the active processes that occur across the cell membrane in order to maintain the state of disequilibrium, and that's largely contributed to the sodium-potassium exchange pump. This is an active process, and the way it works is the sodium-potassium exchange pump is going to eject 3 sodium for every 2 potassium. So it's going to kick 3 sodium out of the cell for every 2 potassium that it brings into the cell. And this is going to help balance this passive forces of diffusion. And it's also going to stabilize that resting membrane potential of -70. So for instance, if you lose, here's your cell again, if you lose 3 positively charged ions and you're only replacing them with 2 positively charged ions, over 1,000,000 rounds of that, the inside is going to be relatively negative. It's going to be relatively negative compared to outside. That's the big difference. So the ratio of sodium entry and potassium loss through that passive channel is about a 3:2. So sodium and potassium are the primary determinants of the potential, of the transmembrane potential. And we saw that sodium-potassium channels can be passive, as in those leak channels, but they can also be active. So the passive ion channels are just leak channels. They're always open. They're always leaking their ions, while the active ion channels are not passive, but they actually require some type of stimulus. So they open and close in response to stimuli. At rest, most of these gated ion channels are closed. There are 3 types of active channels. We have chemically gated ion channels, voltage gated ion channels, and mechanically gated ion channels. Chemically gated ion channels require some type of key. Something is required to actually open the channel. If we look at this image, up here we see the channel in purple, and there is a binding site for ACH, and recall ACH is acetylcholine. So acetylcholine here is the key, and when acetylcholine binds its receptor, right here in the middle, we can see that that opens up that particular channel. When ACE is not present, as we have down here at the lower part of this image, it is closed. So chemically gated ion channels require some type of key. And in this case, the key is a chemical, and the example that we used was acetylcholine. So this is found on cell bodies and dendrites within neurons. Voltage-gated ion channels respond to changes in that transmembrane potential. Here we see changes from -70 to -60 to even +30. So changes in the transmembrane potential as it moves from -70 to -60, we get the voltage-gated ion channels open. So it requires a change in the voltage in order to open or close these particular channels. So they respond to changes in membrane potential. These are found in the axons of neurons, as well as the sarcolemma of skeletal and cardiac muscle cells. Activation gate opens when stimulated, and the inactivation gate closes to stop the movement of ions in or out of the cell. And there's 3 possible states, as we already noted: closed but capable of opening, open, which we refer to as activated, or closed and incapable of opening, which is inactivated. The last type of channel is the mechanically gated ion channel. This is going to respond to some type of distortion in the membrane itself. In fact, up here at the top of this image, we see the ion channel is closed, but if you apply pressure to one side of that channel, it's going to force that channel open just from the applied pressure. And then once that pressure is removed, we can see that the channel closes again. So this is found in sensory receptors that respond to touch or pressure or vibration. So the applied pressure could be a touch, You can press down on skin, and the pressure is going to open up these ion channels to allow ion passage, and then once that pressure is removed, that ion channel closes. So from rest, we experience graded potentials. Remember, these are temporary, localized changes in transmembrane potential. So they can't spread far from the site of stimulation. If you were to consider your axon again, if a graded potential is initiated right here, it's only going to affect this area. It's not going to spread further down the length of the axon. And that is what we mean by localized. It's only going to affect the local area. Graded potentials are produced by any stimulus that is going to open a gated channel. So you need some type of key, such as acetylcholine, to open a gated channel. And once a resting membrane is exposed to a chemical key, the chemically gated sodium channels are going to open. Sodium is going to rush into the cell. And again, I need to ask, what is driving the rush of sodium into the cell? Well, this again is where we have those two gradients. So chemically, sodium wants in order to reach a state of equilibrium. It wants to equilibrate inside and outside of the cell. So that is the chemical gradient. Secondly, you have an electrical. An electrical gradient. Sodium, which is positively charged, is highly attracted to the negative 70 millivolt reading that is found on the inside of the cell relative to out during rest. So chemically and electrically, as soon as that gated sodium channel opens, sodium is going to flood in. And as sodium floods in, we get a depolarization. So let me draw a scale here, and this is going to be rather crude. We'll start with negative 90 millivolt, and we have negative 70 here at rest. We have 0, and then up here at the top we'll have plus 30. So these are transmembrane potential, and these are in millivolts. So if we have a cell at rest, it's sitting here at negative 70 millivolts. As, for example, acetylcholine, if it binds that chemically gated sodium channel, it's going to open that channel. Sodium is going to flood in. As positive charges flood into the cell, it's going to cause this movement up the scale. So we become less negative. That is what depolarization is. It is a membrane potential in which one becomes less negative. And as we go from negative 70 to a less negative number, as we depolarize that cell, that is what we refer to as a graded potential, a temporary, localized change in the transmembrane potential from negative 70 or rest to a less negative number. Sodium ions are going to move parallel to the plasma membrane, producing a local current, which depolarizes the nearby regions of the plasma membrane. That is our graded potential. It's a change in the potential is proportional to the stimulus. So if the stimulus is small, the change in that potential from negative 70 to maybe negative 69 is small. That is graded potential. Here we have a figure that kind of represents that. Here at the top of this image, we have our resting state. And again, at rest, you should think negative 70 always. So our membrane is at rest. It is at negative 70 millivolts. You can see that these ion channels are closed. The first step here is stimulation. The membrane exposed to some type of chemical, such as acetylcholine, that causes an opening of the sodium ion channels. As soon as acetylcholine binds, it's going to open this active sodium channel, and sodium is going to rush in. Again, why is sodium rushing in? Because there is both a chemical gradient pushing the equilibrium of sodium. So sodium wants in to reach a state of equilibrium, but it's also going to be driven by the electrical gradient. Sodium is positively charged and it wants in because it is negative inside relative to out. And we know that opposite charges attract. So as sodium floods into the cell, it causes a change in the transmembrane potential from -70 to -65. That is a depolarization, a change from a less negative number to a less negative number. Depolarization, we are depolarizing this segment of the cell. Step 2 is the graded potential. You're going to get a spread of sodium ions. As sodium floods into the cell, they're going to spread in each direction, okay, along the inner surface, producing a local current that is going to cause a depolarization of the adjacent portions of the plasma membrane. And we can see that here happening. So here, this segment of the membrane is at rest. Here we have a slight depolarization and then a larger depolarization. So that's a local current. Let me just step back and say something else. If this is nothing more than a graded potential, if it's not converted to an action potential, then this all stops. And the sodium-potassium exchange pumps are going to kick the sodium out, and the transmembrane potential of the cell is going to revert to -70. And that is what a graded potential is. It's a temporary, localized change in the transmembrane potential. So it does not last long. Graded potentials, when the stimulus is removed, so we're thinking acetylcholine, the key, the membrane potential is going to return to normal. We call that return to normal, or the return to a more negative number, a repolarization. So remember, depolarization is a movement to a less negative number. So if we had our scale, and I'm just going to have to interrupt this image. So here's -90 millivolts, here's -70 millivolts, here's 0, and here's 30. Okay, if we're sitting at rest, a depolarization is a movement in this direction, and a repolarization would be a movement back towards -70. There are cases in which we also have a hyperpolarization. This is results from the opening of potassium ion channels. Positive ions move out, not into the cell, and it's going to have an opposite effect of the openings of your sodium ion channels. So you get an increase in the negativity of the resting membrane potential, and that is where we get this -90 from. So from -70 to -90 is a hyperpolarization. You're going to increase the negativity of the resting potential. And we'll see how this applies later. So characteristics of a graded potential. The membrane potential is most changed at the site of stimulation. Okay, so if you think about a neuron, if the site of stimulation is right here, that is where most change is going to occur, in that little region of the cell membrane. But the effect can spread passively due to local currents. It can just spread to adjacent segments or areas of that cell membrane due to the local currents. Graded change in membrane potential involves both depolarization or hyperpolarization, and the stronger the stimuli, the greater the change in that membrane potential, so it affects a larger area.